So welcome back from your break. Let's take a look at the concept check question, and then we'll talk about the seedless vascular plants, which is the last section for this recording. So how does the life cycle of bryophytes differ from other plants? Well, remember, it is the bryophytes, the hornworts, the liverworts, and the mosses that have a life cycle that is dominated by the haploid stage or the gametophyte stage. That is totally unique to the bryophytes as a group of plants. Their life cycle is gametophyte dominant. The haploid stage is the part of the life cycle that you see when you see these plants in the environment. So when you see a moss growing on the side of a tree or on a rock or something like that, you are looking at the gametophytes. If you get down and look very closely at the right time of year, you may actually even see the sporophytes, but they are very small, very reduced in size, and completely dependent on the gametophyte. But we're about to see that change as we transition to the seedless vascular plants. So for the first period of the colonization of dry land by plants, the landscape was totally dominated by bryophytes and plants that were similar to bryophytes. And this period may have lasted for somewhere around 50 to 100 million years, somewhere in that time frame. Vascular plants start to emerge during the Devonian, and the seedless vascular plants come to dominate the landscape in the Carboniferous. So in the late Devonian and throughout the Carboniferous, we start to see the emergence of the first ecosystems that we would describe as forests, but they're not the sort of forests that we would recognize today, because these are forests that are made up of lycophytes and ferns and their relatives. So these would be forests that look completely different, but they also represent a time period in the Earth's history where a large amount of the fossilized plant material that we use as fuel, so the peats and the coal, are formed from plant material that would fall into large bodies of fresh water that is oxygen depleted, and therefore very little bacterial activity is taking place. And so this material, rather than being degraded by bacteria, would be overlain by sediment, more plant material, and over time it would be covered by substantial amounts of pressure or substantial amounts of sediment and subject to a lot of pressure and heat, and would slowly fossilize, and the organic matter would be converted into peat and then subsequently into coal. Hence the name the Carboniferous, because these are fossilized or carbon-based fuel sources. And this is the time period when those large deposits of coal and peat that we still use as sources of energy were laid down. And it is due to the lycophytes and the manilophytes and the first forests that they would form. So when we start looking for the earliest of the fossils that might be precursors to vascular plants, we look back around 420 million years ago into the fossil record. And there we see plants that look a little bit like this. Now, what tells us that these are likely early vascular plants is the fact that the stems, so let me fire up the pen again. So the evidence for vascular tissue. Vertical stems. Because vascular tissue contains a second polymer called lignin. And lignin provides secondary thickening to the cell walls and therefore allows the cells of the vascular tissue and cells associated with the vascular tissue to provide vertical support against the force of gravity. So the fact that we're starting to see vertical stems means that this is evidence for early vascular tissue. The second part is bifurcated stems. Meaning that the stems are splitting and you get two branches coming off each split point. So you've got two branches there, you've got two branches there, we've got two branches there. The third piece of evidence is sporangia at the tip of each stem, which means there is some sort of nutrient support that allows for the growth of the sporangia, or improved nutrient support, improved mobilization of material up these stems to support the growth of sporangia on the tips of each stem. The final is rhizoids that are capable of mobilizing water and minerals. Now, it would be a mistake to suggest that these rhizoids are true roots at this point. They're probably not. They probably don't contain fully developed vascular tissue, nor would these stems contain fully developed vascular tissue, but they show the beginnings of vascular tissue. So these are the sort of plants that we start to see in the fossil record around 420 million years ago, letting us know that the very earliest forms of vascular tissue are starting to appear. What is also interesting is that we see the appearance of stomata. Now, you may recall from the taxonomy lab that stomata do appear in the moss, but not in the hornworts and the liverworts. But we do see the re-emergence or the appearance of stomata as regular features in these vertical stems with the appearance of these fossils. And so that also gives us a hint that we are starting to see the emergence of vascular plants and vascular tissue, because stomata appear to be part of the physical features of vascular plants, because all modern vascular plants have stomata. And that stands to reason because stomata are important for gas exchange. They also allow for water to be lost through transpiration. So if the stomata are present and you're going to lose water through them, then you need to be able to mobilize water more efficiently. That means vascular tissue. So the appearance of stomata also hints at the emergence of vascular tissue. So these are all important features that hint that these plants around 420 million years ago are starting to show very early vascular tissue. So the types of vascular tissue that modern plants have is called xylem and phloem. Xylem mobilizes water and carries water from the roots to the body of the plant. Oops. Keep remembering to fire up the pen every time I change slide. So transports water. So that's the role of xylem. Phloem carries photosynthates. So these would be the products of photosynthesis from leaves to roots, shoots, and developing stems. So not all leaves that are present on a plant can actually carry out photosynthesis sufficiently to support not only themselves, but the rest of the plant. So developing stems where the leaves are still small, still just beginning the process of photosynthesis, are still getting support from the larger leaves lower down on the plant that are fully developed and are actively carrying out photosynthesis. Once you see the emergence of xylem and phloem, vascular tissue, then you can start talking about the presence of roots, shoots, and leaves, because each of those is defined by the presence of true vascular tissue. So the other feature that we now see that is a change in the life cycle is the fact that with the emergence of vascular tissue, the sporophyte now becomes the dominant generation in the life cycle, not the gametophyte, because now you have the capacity to mobilize large volumes of water, large volumes of minerals, and the products of photosynthesis to allow for the growth of the plant to something that is larger. And with the presence of vascular tissue, you now have the capacity for the plant to grow vertically, because it now has a means of supporting itself against the force of gravity. And so the sporophytes now become quite large. On the other hand, the gametophytes are still present as a distinct structure, but they are completely independent of the sporophyte. So you now have an independent sporophyte and gametophyte. Turn on, make sure the pen's on. Actually, I shouldn't say individuals. It helps if I use the eraser before I take it down. What I should say is two separate multicellular stages. So remember again, the bryophyte life cycle is characterized by the fact that the sporophyte is completely dependent on the gametophyte. It is small, it's reduced in size, it is more or less incapable of photosynthesis. So it depends upon the gametophyte for its nutrition. Here we now have the gametophyte and the sporophyte present as two separate independent multicellular stages. But the one that is larger now is the sporophyte. In fact, fern gametophytes, as a rule, are smaller than your pinky finger fingernail, probably about half the size of your pinky finger fingernail. If you want to find them, you can go out where you see ferns growing in the landscape. If you get down on your hands and knees under the ferns at the right time of the year, you'll see these tiny little flat-looking green structures sitting on the surface of the soil. That is almost certainly the fern gametophyte generation of the ferns. Those will eventually give rise to the new sporophyte, because fertilization is still going to take place within the body of the gametophyte, and the sporophyte will initially appear to be growing out of the gametophyte. But in fact, it is not getting any support from the gametophyte at all. Once it develops its own leaves, it begins photosynthesis and supports itself, and the gametophyte shrivels up and disappears, so that the sporophyte lives a separate existence from the gametophyte. And the two actually maintain an independent existence from each other. So if we take a look at the life cycle, we've now recognized that the sporophyte is the dominant form in the landscape, so we might as well focus on that. So here's our sporophyte. And notice that a big deal is being made out of the leaves here. These leaves are sporophylls. Well, actually, I should be a little bit more specific here. So the leaf, and I'll do it in, well, actually, no, I won't do it in green. I'll do it in a different color. So this is a compound leaf. And what color? Let's do it in blue. That is called a leaflet. So on the underside of the leaflets within the compound leaf, you have the sporangia in a structure called a sorus, and this is classic fern structure. So what we now need to do is introduce a new term. The term is sporophyll, and its definition is a leaf modified for reproduction. This is the first time we've encountered it, but we'll encounter sporophylls from this point onwards as part of the reproductive process of all plants. Because surprisingly enough, all reproductive structures from this point onwards that we'll see in the plants are based on sporophylls, leaves modified for the reproductive purpose. So when you look at the blades of a pine cone, each one of those blades is actually a sporophyll. It's a modified leaf. If you look at the petals on a flower, those are sporophylls, leaves modified for the reproductive process. And in fact, there are parts of a flower that really do look like leaves. So the petals, the sepals, the green leaf-like arrangements that actually cover the flower bud. But what you may not realize is that the pollen-bearing part of the flower, the stamens, and the central female part of the flower, the carpel, are actually sporophylls too. They are derived from leaves that have been modified for reproduction. So all reproductive structures from this point onwards are actually constructed by modified leaves, sporophylls. So this is another term that's really important to understand. So here we see the sporophyll as it emerges in both the lycophytes and in the manilophytes, the ferns. So in lycophytes, you have structures called strobili or strobilus for the singular. That is, again, built out of layers of sporophylls, leaves modified for reproductive purposes, and it looks very much like a cone. So in fact, a pine cone is more correctly referred to as a strobilus rather than as a cone. A cone-like structure is actually a strobilus. So we see them in lycophytes, we see them in the conifers. So within the sporangia, coming back to our fern life cycle, within the sporangium, you have sporocytes that are going to undergo meiosis. That gives rise to the spores. The spores are then released, coated in their sporopollen coat. Those land on the ground. If the conditions are conducive to germination of the spore, it germinates and it begins dividing by mitosis to give rise to the gametophyte. Here, there is a single gametophyte. So, single, that's not really good. Excuse me. So we have a single gametophyte. Containing both gametangia. So both the antheridium and the archegonium are contained within this single gametophyte. And so here we now see another advantage that ferns particularly have over the non-vascular plants. Remember that in the non-vascular plants, the bryophytes, the gametophytes were two separate structures, antheridium on one, archegonium on the other, and the sperm had to swim a substantial distance to get from the antheridium to the archegonium, which required a substantial amount of water for that to take place, which restricted the bryophytes to moist environments where there was a substantial amount of water. Ferns don't require that. They still have a flagellated sperm, so they still need water. But given that the gametophyte is small and contains both gametangia, the amount of water that's required could be as little as a single drop of rain or a single drop of dew to provide enough water for the sperm to move from one gametangium to the other gametangium. This gives an added advantage to the ferns and to some degree the lycophytes as well, because now they can penetrate drier soil. And so they're not restricted to moist habitats. They can now penetrate into drier habitats because they can mobilize more water out of the soil. They can mobilize it over longer distances because they've got vascular tissue. They can grow up because they've got vascular tissue, so they can get large and support themselves against gravity. And they have a gametophyte that contains both gametangia, so they only need a very small amount of water for reproductive purposes. A single drop of water, raindrop or dewdrop, can do the task. I'm going to put an arrow down here so I can utilize this. One raindrop or dewdrop is sufficient. So again, this gives them a real advantage. They can now move into environments that contain substantially less surface water because they can mobilize water out of the soil and they don't need very much water for reproductive purposes. So the sperm swims from the antheridium to the archegonium, just as we've seen previously. It enters the archegonium, fertilizes the egg cell. The zygote starts its life inside the archegonium. The sporophyte grows out of the archegonium and develops leaves, which allows it to begin photosynthesis. It grows a stem, which is referred to as a rhizome. So that's another feature that we need to throw in here. Let me put this up here so I can utilize the bottom. So this, oops. So this thing is called a rhizome. And this is a horizontal stem. And it's often underground. Or right on the surface. So you can see the body plan here of these sorts of plants is a little bit different. So you've got the horizontal stem with its roots, mobilizing water and minerals, and then the leaves, which we call fronds on a fern, which are really compound leaves, grow out of this. And that's what we see growing in the landscape. It's actually the fronds, the compound leaves that we see mostly in the landscape. The stems typically are right on the surface of the ground, maybe extend a little bit above the ground. There are ferns that have vertical stems, a group of ferns called tree ferns, which can grow up to about 6 feet in height, have a rather larger stem that is more vertical than it is horizontal, but the fronds are quite large on these ferns as well. So the life cycle is different. Here we've seen a separation between the gametophyte and the sporophyte. No longer does the gametophyte dominate the life cycle. It's the sporophyte that dominates the life cycle, but we still have a gametophyte. It's just small, very reduced in size, and the sporophyte and the gametophyte are separate from each other. And this is the classic seedless vascular plant life cycle as exemplified by ferns. So the two life cycles that you need to know out of this chapter are the moss life cycle, which we've already talked about, and the fern life cycle, which we just finished talking about. Those are the two life cycles that you are responsible for in this chapter. You should also know the generalized life cycle of plants, that very first little life cycle that I introduced you to at the beginning of this chapter. So when it comes to the two types of vascular tissue, the xylem is functional when the cells are dead. These are hollow tubes that carry water and minerals vertically up the stem. They begin, we think, from a group of cells that are shaped like this, called tracheids, and these overlap with each other and allows the water then to move through the cells like this, through openings through the cells, through the plasmodesmata in the cell walls, the openings that contain the plasmodesmata. And the water moves up through that. They are reinforced by lignin. So the cell walls are reinforced let's remember to erase before I go back to the pen. So lignin acts as a reinforcement. It gives rise to what is called secondary cell wall thickening. And I'm just going to put here probably derived fiber cells, because fiber cells look exactly the same, but they are living cells until they die. And then in some cases, fiber cells, well, if you think in terms of traditional ropes made out of hemp and other material, nut shells. So if you're trying to get into an almond to shell it, or a pistachio, or a walnut or pecan or something like that, when you're trying to crack through that hard nut shell, that's all made out of fiber cells. The other place you might find them, if you've ever eaten a pear, you know that a pear, the pear has a gritty texture. That's actually fiber cells embedded in the flesh of the pear. So these are all cells that are referred to as fiber cells. They have this secondary cell wall thickening made out of lignin. They look very much like the tracheids. They have this same elliptical shape, but in the xylem that is derived from the tracheids, these are dead cells that allow the water to pass through the plasmodesmata. In the case of the phloem, the cells are elongated cells that are butt-ended to each other. They are living. they usually have a second cell next to them called a companion cell. So the companion cell supports the phloem cell. Because the one thing that a phloem cell does not have is a nucleus, but it has everything else. So the companion cell has the nucleus, and we know that the companion cell and its adjacent phloem cell are actually the product of mitosis. So they are the two daughter cells of mitosis. One loses its nucleus and becomes the actual phloem cell that is involved in the transport of sap, the photosynthate, and the companion cell retains its nucleus and supports the phloem cell. So these are living cells. They just don't have a nucleus, but this companion cell provides the nuclear support, the genetic support, I guess, the DNA support for the cell. And it's the development of these two types of transport cells that allow vascular cells now to, vascular plants to get really big and to grow very big. And we see that exemplified in the fossil record of the early vascular plants, which we'll talk about in a moment. So in terms of the evolution of roots, it's not entirely clear where roots evolve from. But what we do know is that when we look at the structure of early roots, and even when we look at the structure of the vascular tissue in modern roots, it looks very much like the structure of early stems, and particularly the base of early stems. So it may very well be that roots evolved as extensions growing from the base of stem tissue. They certainly have an independent growth from the stems. Remember, both roots and stems grow from the apical meristem tissue that's located at the tips of the roots and the stems, not at the base of the roots and the stems. So they have an independent growth. But however the roots evolved, they do grow from the base of the stem. And so we think that they may have emerged as extensions of early stem structures. In the case of leaves, we recognize two different types of leaves, microphylls and megaphylls or macrophylls. So microphylls are found in lycophytes only, and megaphylls are found in all other vascular plants. So the difference between a microphyll and a megaphyll or a macrophyll, some people say macrophyll, some people say megaphylls, so you'll see both terms utilized, is the nature of the vein system. So in a microphyll, there is a single vein that supports a small amount of photosynthetic tissue. A megaphyll has a highly branched vascular system. So the classic vein system of a leaf that hopefully you're familiar with, that is highly branched, that's a megaphyll. And that's what we see in all vascular plants other than lycophytes. So a microphyll almost certainly evolved as an extension of the stem of early vascular plants, where a single vein branched out from the stem and was surrounded by photosynthetic tissue that spread out in a flattened surface to maximize light collection. And that, we believe, is the origin of microphylls. Megaphylls may have arisen from microphylls that branched as a consequence of expanding the vascular tissue in the early ancestors of all vascular plants other than the lycophytes. We're not entirely sure what the relationship is between lycophytes and all other vascular plants in terms of their evolution. It's not entirely clear, but megaphils probably evolved from a branched extension of the stem that was surrounded by multiple layers of flattened photosynthetic tissue, because that's what we certainly see in those vascular plants that we consider to be basal to the ferns and other vascular plants other than the lycophytes. But exactly how those megaphils evolved from the microphils, we're not entirely sure. But there is some suggestion that, in fact, microphils may represent a different evolutionary step to the megaphils, that, in fact, the evolution of leaves as microphils and megaphils may actually represent two separate evolutionary steps. That microphils represent one evolutionary pathway that gave rise to the lycophytes, and megaphils represent a second evolutionary pathway that gave rise to all other vascular plants. Again, the relationship between lycophytes and all other vascular plants is not clear. But if this is in fact the origin of microphils versus megaphils, that would be consistent with lycophytes emerging, or the ancestors of lycophytes branching from the common ancestor of all vascular plants before the evolution of leaves. And then you have two separate evolutionary pathways leading to leaf structures, the microphils versus the megaphils. So I've talked a little bit about sporophylls, the fact that these are leaves modified for reproductive purposes with the presence of sporangia giving rise to spores. So you've got the sporophylls of the ferns with their soroid, then you've got the strobili, the cone structures that we see in lycophytes and also in gymnosperms as cone-like structures being formed out of sporophylls. One of the other things that we should also point out is that in seedless vascular plants, there is only a single type of spore that gives rise to the gametophyte that is bisexual. That is, it contains both the female and the male gametangia. The vast majority of seeded plants, however, have a megaspore and a microspore. They are what are referred to as heterosporous, meaning they have two different types of spores, a female spore and a male spore. And as a consequence of that, you have a female gametophyte and a male gametophyte. There are some ferns, most notably the water ferns. So I'll put that in here. Which are a group of seedless vascular plants that are actually heterosporous. So they are unique amongst the seedless vascular plants in that they produce two different types of spores. They are heterosporous. Why this is important is because we know that heterosporine is required for the evolution of seeds. Why can we say that? Because there are no plants extant today that are seeded plants that are not heterosporous. In other words, there are no homosporous seeded plants, nor are there homosporous seeded plants in the fossil record. Where we can clearly identify seed-bearing plants in the fossil record, they are all heterosporous. So maybe I should make a note of that. So the fact that you can't have a homosporous seed-bearing plant strongly suggests that being heterosporous is a condition necessary for the evolution of seeds. So we can summarize it in this diagram, and this comes straight out of the textbook. There's a version of this in the textbook. Homosporous vascular plants produce a single type of spore that produces a bisexual gametophyte. So I'll put in here contains both archegonium and that's what we mean by bisexual. So in the heterosporous plants, you have a female gametophyte with an archegonium. Up to a point. We do lose the archegonium and the antheridium. As seed plants evolve. So in the transition from gametophyte, from gymnosperms to angiosperms, we actually lose the archegonium and the antheridium. In fact, the antheridium pretty much disappears in the gymnosperms. The archegonium hangs on in the gymnosperms, but even it disappears when we transition to the angiosperms. But in those plants that are heterosporous, that are more basal amongst the gymnosperms, there is both an archegonium and an antheridium still in the gametophytes. So this is something that we are going to see as part of the ongoing adaptation to life on dry land, because the production of seeds is considered to be an adaptation to success as a colonizer of dry land, as you'll see in the next chapter. So let's take a look at the two clades of seedless vascular plants. They are the Lycophyta, otherwise sometimes known as the Lycopodophyta. In here, aka Lycopodophyta. These are the club mosses, spike mosses, and quillworts. And as I mentioned earlier, unfortunate choice of terms because they are neither mosses nor worts, but these are the common names that are given to them. And then you have Manilophyta, the ferns, the horsetails, and whisk ferns. As I mentioned back at the beginning, these were in the past assigned to 3 different phyla. I will give you the names of the 3 different phyla, but we do recognize them as 3 members of the same clade now, Manilophyta, on the basis of DNA sequences and some biochemical and physical features. So the Lycophyta are the first to emerge. They are survivors of the Devonian, and along with the ferns, form the first true forests that dominate the Carboniferous landscape. They can be quite large in some cases. So when we take a look at the representatives, here they are. So Selaginella is a spike moss. Not entirely sure why they're referred to as a spike moss, because they don't look particularly spiky, but that's the name they've been given. You can understand why a quillwort is called a quillwort, because it does look like a bunch of quills. And then the club moss is so named because of the presence of this cone-like structure, the strobili. Remember, the strobilus is a product of layers of sporophylls, leaves modified for reproductive purposes. So the sporangia of the club mosses are located within the strobili on the sporophylls. When we look at Manilophyta, these are the ferns and their relatives. So Xylophyta, xylophytes, is the old phylum name for the whisk ferns. And the reason why they were not considered to be true ferns for the longest time is because they have the bifurcated branching. That's what's meant by dichotomous. It's just another name for bifurcated branching, so splitting into two. They don't have leaves and they don't have roots in the formal sense of the term. So they look like they may have evolved before vascular tissue came to dominate the plant body. But once we take a look at DNA sequences and we take a close look at the structure, that's what's meant by ultrastructural details, using an electron microscope to look deep into the structural relationships of the plant tissue, what we realize is that they are actually descended from an ancestor that did have vascular tissue and possessed true roots and true leaves. So the loss of the roots and leaves as typical structures are considered to be secondary features. So these are the loss of true roots and true leaves as part of the evolution of these from an ancestor that did have roots and leaves. Sphenophyta is the old phylum name for horsetails. And horsetails are so named because they have one of the two growth forms, because they have two growth forms, a vegetative growth, which means no reproductive structures, and then a reproductive growth, or a growth that contains reproductive structures. And in the reproductive structure, they have a bushy appearance, which is why they are referred to as horsetails. It's a single L. I've been putting, let me fix that here, two L's for some reason. So the old phylum name for ferns is Pterophyta. So phylum Manilophyta is the combination of Pterophyta, the ferns, Sphenophyta, the horsetails, and Xylophyta, the whisk ferns. Sorry, I'm still having a little bit of a anyway, so here you can see representatives of all of them. Here is a true fern, horsetail. You can see the bushy growth. So in fact, the bushy growth is vegetative growth, not reproductive growth. I was right the first time. And then the whisk ferns, and you can see there's no indication of true leaves. You're not going to find true roots underneath. And you have the sporangia on the tips of these bifurcated stems. So it really does look like the fossil that we were looking at earlier that gave us the hint that there was the emergence of vascular tissue in the fossil record. The whisk ferns look very much like that. So that's why these were considered for a long time to be living fossils. Living fossil being a term that is poorly chosen, that refers to an organism that appears to have remained essentially unchanged for long periods of evolutionary time, based on a comparison of fossil evidence versus the extant organism. The problem with using the term living fossil is that the fossil usually does not preserve subtle changes that may have occurred. So in the case of an animal, for instance, with hard parts, all it preserves is a skeleton, but it doesn't show you any evidence of changes in external appearance or behavior or anything like that that may have created a different species. So we still use the term living fossil, even though it is very hard to defend a term like that in many instances. So whisk ferns were considered to be living fossils for a long period of time until DNA and deep level microscopic analysis or ultrastructural analysis indicated that they really are true ferns and belong amongst the ferns, along with the horsetails. So what we see today amongst the horsetails, particularly, and some of the lycophytes, is I'm trying to think of a poor representation of what they were truly like once in the distant evolutionary past. In the fossil record, we have horsetails that were 15 meters, and if you're not sure what 15 meters represents, that's roughly 50 to 60 feet high and about 6 feet in diameter, based on fossil evidence. These were truly large. These were tree size in every sense of the word, along with ferns that were more tree in appearance, lycophytes that were more tree in appearance. These are just what we have today is a poor representation of what these were like in the past. The average horsetail today is about 3 feet tall and maybe the thickness of a large pencil. And they grow in marshy habitats, in the drainage ditches along the sides of roads, along the sides of trees, of streams. What is interesting is that they have, in the vegetative growth, a hollow stem. And the reason why they're hollow is because it allows oxygen to get down to the roots. Because they're growing in marshy habitats, there's not a lot of open air space in the soil to allow air to penetrate to carry oxygen to the roots. In fact, this is the reason why you can kill your potted plants if you overwater them. They'll literally suffocate because the water blocks all the air spaces between the soil grains and prevents air from getting down to allow oxygen to get to the roots. Water is a poor conductor of oxygen because oxygen does not dissolve terribly well in water. And so because the horsetails are growing in very marshy habitats and the roots could potentially suffocate, the stems actually have a hollow straw-like appearance, and that allows the oxygen to get down to the stems and support the stem growth. So an interesting adaptation to the sorts of environments that they're growing in. So that's, yeah, it's mentioned there. I couldn't remember whether I put it in notes. I wanted to remember it. It's there in the notes, so you don't have to worry about that. And again, the reproductive stems have the strobili, these cone-like arrangements built out of the sporophylls that contain the sporangia that produce the spores. Ferns also become quite large in the Carboniferous forest. Here, the term tree fern really does refer to a fern that is the size of a tree. These can be upwards of 100 to 150 feet in height, and they have a branch structure that looks more like a tree than a fern. We have some fossil evidence for this, and in fact, we point to some of these as potential ancestors of gymnosperms because they show evidence of potentially being heterosporous and potentially giving rise to the two gametophyte structures that we know are essential for the evolution of leaves. of seeds. And this is speculative, it's not definitive, but the fossil evidence suggests that this may be a potential route to seeded plants. We don't know for sure. Again, as we mentioned before, Lycophyta have the microphyls, and only Lycophyta have microphyls. All other vascular plants have the megaphylls or macrophylls, depending on how you want to name them. You'll see both names used. Ferns, of course, have compound leaves, or modern ferns have compound leaves that roll out from what's called a fiddlehead, which is a rolled up leaf, looks like the head at the end of a violin, which has that scrolled appearance, rolled appearance, hence the name fiddlehead. And there's actually a fern called a fiddlehead fern that retains that scrolled appearance, that rolled up appearance, once the compound leaf completely emerges. The very end of it still remains rolled up. The vast majority of ferns are homosporous, with the exception of the water ferns, as I mentioned earlier. And we've talked about the requirement for a small amount of water because the sperm only has to swim over a short distance. I've basically filled in all of this with the life cycle. So when we start thinking about the significance of seedless vascular plants, it's their role in forming the first true forests in the Carboniferous that makes them incredibly important. This is the source of our fossilized plant fuels, so the peat and the coal. But what this also does is increase the levels of oxygen in the atmosphere, along with cooling down the planet, because they're subtracting a lot of carbon dioxide out of the atmosphere. This would lead to some animals, arthropods, and particularly insects, experimenting to see just how big they can get, because the oxygen level here gets quite high. The current atmosphere contains about 20% oxygen. Here we think the oxygen levels may have risen to as much as 35% oxygen. Why that's important to arthropods, and particularly to insects, is because they use features called trachea, and you'll see those when we do the animal kingdom later in the semester, that lead into tubes that, well, the tubes are called trachea, the openings are called spiracles, and they penetrate into the body of the insect. Why? Because the oxygen diffuses into the body of the animal. And so you can't have the tissues and cells of an insect more than a few cells away from direct contact with the air, because diffusion only works over short distances, even in gas phase. And so increasing the level of oxygen in the atmosphere increases the distance over which the oxygen can diffuse into the body of the insect. And so the insects actually start to get quite large. This is an era when dragonflies, and you can see a picture of a dragonfly or a distant relative of the dragonfly here. These may have had a wingspan of about 3 feet. These were large insects. Centipedes, and millipedes, which were some of the first arthropods to actually permanently live on dry land, may have reached 6 feet in length and 1 foot across. Very large arthropods, all because of this huge amount of plant growth in the form of horsetails, ferns of different types. So these trees that you see here, these are ferns. Here you can see lycophyte. Looks like a large club moss, in fact. You've got a large horsetail here. So these are all members of the first true forests. And so you have an ecosystem that doesn't look anything like an ecosystem that we might be familiar with today, but we still use the same terminology. It's a forest, we've got trees, we've got arthropods, but we've got arthropods probably of our worst nightmares, and trees that look nothing like anything that we would be familiar with, because they are in fact horsetails and ferns and other plants that are not really trees in the sense that we think of them. This is why these are so important. So one more concept check, 2 questions, and then we're done. So what are the key derived traits found in manilophytes and seed plants, but not in lycophytes? And how do the main similarities and differences between non-vascular plants and seedless vascular plants affect the function of these plants? So key derived traits of manilophytes and seed plants that are not found in lycophytes. That would be the presence of microphylls in lycophytes versus megaphylls in all other vascular plants, and the highly branched nature of the vascular system in seeded plants and manilophytes versus that in lycophytes. Why are they key? Because they allow for these plants to get truly large. Lycophytes got large, but manilophytes and seeded plants got substantially larger than lycophytes ever got. It gives them an advantage in the landscape. They can mobilize more water, they can mobilize more minerals, they can mobilize more photosynthates, they can get truly large, they have well-developed support against gravity. It allows them to really start to dominate the landscape and leads to the first true forests in the sense that we think of them, and later forests in terms of seed plants. Also remember that in manilophytes, we start to see the emergence of the heterosporous condition, and that certainly becomes part of the makeup of all seeded plants. It is what leads to the evolution of seeds in the first place. The development of the heterosporous condition. How do the similarities between non-vascular plants and seedless vascular plants affect the function of those plants? Well, the 2 similarities versus differences. They both have flagellated sperm for reproductive purposes. So that would be a key similarity. But bear in mind that in non-vascular plants, so the bryophytes, the sperm are produced on a gametophyte that is at some distance from the gametophyte that contains the archegonium that produces the egg. So this restricts the non-vascular plants to very moist environments where there's substantial amounts of water. In the case of vascular plants, seedless vascular plants, remember the 2 gametangia, the archegonium and the antheridium, are located on a single gametophyte. So small amounts of water are necessary, so they can reproduce successfully in much drier landscapes because they only need a single drop of water. So that's one difference versus similarity. The other major difference is that non-vascular plants don't have vascular tissue. They have to rely on diffusion only. And so they have to remain relatively small and close to the ground because diffusion only works over a short distance, 2 to 3 cells. Vascular plants have vascular tissue. So they now have roots that can mobilize nutrients and water from the soil. They have vascular tissue in the stems that can transport that over large distances vertically and also provide support so that they can grow vertically. And they have vascular tissue that can provide support for photosynthetic tissue so that it can expand out and form large flat surfaces. We call them leaves, for improved light gathering to allow for greater levels of photosynthesis, something that the non-vascular plants can't do. Do they have root-like structures? Yes, they're called rhizoids, but they're for anchoring to the soil, not for mobilizing water and nutrients. Do they have stem-like features? Yes, but they are very short, with the exception of mosses that can have slightly larger vertical features because they have conducting tissue, but most bryophytes don't have that. Do they have leaf-like features? Yes, but they are incredibly small because, again, they are reliant on diffusion for mobilizing the products of photosynthesis to the rest of the plant body, so they can only be small. So that's how the similarities and differences between non-vascular plants and seedless vascular plants can affect the function of these plants as photosynthetic organisms. So that's it for plants, or at least for the first chapter related to plants. We will obviously cover the next chapter next time. So until then, be safe. If you have any concerns, questions, or comments about anything that I've covered here, be sure to come and see me during office hours or shoot me an email, and I'll be more than happy to help you out, clarifying anything that you're uncertain about. See you next time.